Sperm morphology assessment looks at the proportion of sperm cells with abnormal shape or morphology against normal cells. Defects in the morphology of a sperm cell, also known as teratospermia, can be classified as head defects, neck and midpiece defects, tail defects, and cytoplasmic droplets. The assessment of sperm morphology is done by microscopic observation of a stained smear of the semen sample. Different staining techniques can be employed. In this video, we'll be using the hematoxylin eosin or H&E staining technique, which is simple, quick, and reliable. For this experiment, you will need the following: freshly collected human semen sample, droppers, glass slides and cover slips, Harris hematoxylin stain, eosin stain, 1% acid alcohol solution, alkaline water or 0.2% ammonia water, 95% ethanol, absolute ethanol, xylene, DPX or Canada balsam, Coplin jars, thumb forceps and pins, compound light microscope, immersion oil. H&E staining is performed as follows. Take a small drop of the liquefied semen sample on a glass slide and make a smear using a second glass slide as shown here. Allow the smear to air dry at room temperature. Immerse the slide in Harris hematoxylin stain for about 20 minutes. Remove the slide and give it 2 to 3 quick dips in 1% acid alcohol solution. Rinse the slide with slightly alkaline water or with 0.2% ammonia water. The stained smear now becomes bluish. Transfer the slide into eosin stain solution and leave it for 2 to 3 minutes. Give the slide 3 to 5 dips in 95% ethanol, followed by another 3 to 5 dips in absolute ethanol with 2 changes, which is then followed by 3 to 5 dips in xylene with 2 changes. Add a drop of mounting medium such as DPX or Canada balsam to the dehydrated and cleared smear. Cover it with cover slip. Allow the slide to air dry overnight and observe under oil immersion objective of the microscope. Study the morphology of 100 to 200 individual sperm cells and assess the morphological defects of the various parts of the sperm cells. Calculate the fraction of abnormal sperm cells in terms of percentage. A semen sample that contains more than 30% of morphologically normal sperm cells is considered normal semen. Abnormal sperm morphology can be caused by a number of factors.